Okay, I'm ready. Hello, everyone, and we're so glad you've joined us for this webinar, The Neurobiology Underlying Drug Addiction, Lessons from Modality Imaging and Neuropsychological Studies in Human. I am the Senior Fellow of LabRoots. I'll be moderating today's session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advocating advancing scientific collaboration and learning. So let's get started. You can pose questions to the speaker any time during the presentation while they're fresh in your mind. To do so, simply type them in the green Q&A box, which will open when you click on the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, please click on the screen icon lower right-hand corner of the slide window. If you experience technical problems seeing or hearing this presentation, please click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at the lower left corner. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your webpage and follow the process to obtain your CE credits. I would like now to present today's speaker, Dr. Rita Goldstein. Nationally and internationally known for her neuroimaging and neuropsychological studies in drug addiction, Dr. Goldstein is a professor of psychiatry with a second appointment in the Department of Neuroscience at the Icahn School of Medicine at Mount Sinai in New York. She is the chief of the Brain Imaging Center, or BIC, and she also directs the Neuropsychoimaging Addiction and Related Conditions research group that uses multimodality functional imaging studies to explore the neurobiological basis of impaired cognitive and emotional functioning in human drug addictions and other disorders of self-control. I will now turn it over to Dr. Goldstein for her presentation. And I think we're having a slight
Okay, so thank you so much for inviting me to this wonderful webinar. I'm going to be talking about the neurobiology underlying drug addiction and the lessons we gain from many years of studying drug addiction using multimodality imaging and neuropsychological tools in humans. Okay, so we all know that many people, millions of individuals, use drugs on a regular basis. Here you can see some very recent statistics showing that about 20 million of individuals are using marijuana, for example, in the past month. These are people above the age of 12. In my lab, we study cocaine, and here around 1.5 million of individuals are using cocaine, have been using it in the past month. Cocaine is also the drug that is only second to heroin in terms of the physical harm and dependence that it causes. And if you look at emergency department visits, that's another indication of how cocaine is really harmful to your health. So here is cocaine #1, exceeding even heroin in terms of the rates of emergency department visits. So around 162 visits per 100,000 individuals. But despite highlighting the severity of using cocaine, not be equivalent or equalized together with bad habits. So just looking at your smartphone every second is not addiction. Drug addiction is not drug abuse, and it's not even drug dependence, as I hinted. So only drug addiction has a constellation of symptoms and presentation that is very unique. It's associated with loss of control over drug intake, over the frequency and quantity of use, despite very catastrophic consequences to the individual's life. to the things that are really important to them, family, friends, job, despite a decrease in the pleasure, in the reward that is gained by the drug. So let me just start with giving an example. For example, a person who is going through a car accident, they're in the hospital, and they're exposed to opioids to decrease pain. That person can become dependent pretty quickly on opioids. You can see it by the fact that you need more and more of the drug, that you have withdrawal effects if you discontinue it abruptly. But it does not mean that the person is addicted to the drug. Only when you start using it beyond what the doctor has ordered, only when it becomes the focus of your livelihood in many ways, only when you lose control over how much and how frequently you use it, despite the fact that it becomes really bad for you, and despite the fact that there is no more pleasure from the drug, this is when we are talking about addiction. So when talking about addiction, it's very helpful to go to biographies and autobiographies, and I have a few of those examples, if I can move the arrow here, to show some of the biographies and autobiographies that are really helpful to read when thinking about addiction. Okay, here it is. And so those are just a few examples of books that are really extremely informative, written very eloquently, and whether about their own life and their own struggles. And those struggles are very, very similar. And I highly recommend those books or any other type of books that you can think about that really introduces you to the concept of addiction. Okay, so how do we think about drug addiction, having described this introduction? So drug addiction is a chronically relapsing brain disorder. It is characterized by repeat drug self-reinforcement, by very, very strong craving sensations that develop with cues that have been previously associated with the drugs, with stress and other type of emotional evocation, and with drugs themselves. Some of the drugs are associated with binges and loss of control over frequency and quantity of use. And they all lead to withdrawal, and withdrawal that is so severe that can contribute to relapse. Although relapse, which is a core part of addiction, going back to using drugs, can happen even after years of abstinence, even when withdrawal symptoms and acute symptoms have subsided. So in our model that we call the iRISA, we emphasize that at the core of this clinical symptomatology of addiction are impairments in response inhibition and Salience attribution. So those are more neurocognitive type of concepts where we are emphasizing how the drug becomes overpotent, it becomes extremely important, and at the expense of other non-drug related cues and stimuli, and with a concomitant decrease in the ability to control behavior and to regulate drug intake. And in this type of thinking about addiction, we also emphasize the importance of the prefrontal cortex and in our ability to regulate our behavior, to inhibit it, to monitor it, to make advantageous decisions. And so taking addiction beyond the type of more classical reward circuit that has been associated before with drug addiction. How do we image that in the human brain, in the alive human brain? Most of the studies of addiction, actually, until today, are done in animals, in small rodents. But our lab is doing similar studies in the alive human brain. So basically, we're very lucky here at the Icahn School of Medicine at Mount Sinai. We have the Brain Imaging Center that adopted a translational approach to imaging the human brain across disorders, including drug addiction. So here we have people who are volunteers. They can be healthy individuals. They can be people with addiction to cocaine, for example. And they come to our studies where we spend a lot of time with them. We interview them. We test them. And we know a lot about their drug use histories. We know that they meet criteria, for example, for drug use disorders, for cocaine use disorder. We know about other alcohol use, other drug use and alcohol use. And we know a lot about their personality and psychological type of profile. So we test their behavior, emotion, cognition, and what people think about as personality, more stable traits, with a whole set of not just interviews, but also paper and pencil tasks and computerized type of tasks. And this is all done even before we put a person in a scanner and to image their brain. So to image their brain structure and morphological integrity or function. And today I'm going to talk about two tools, and we have several to choose from depending on the question, but the results I'm going to present today are from the EEG, and I assume many of you know about the continuous electroencephalography. So this is just basically electrical activity from the scalp. People go, for example, to diagnose seizures sometimes, and they have all these electrodes. And we use those for different purposes. And I'm going to also talk about MRI scans to scan the morphological integrity of the brain, but also its function. And I'm going to say a few words about that a little bit later. So before going to actually prediction of addiction, to say a few more words about our subjects. Our subjects are mostly cocaine-addicted individuals who meet criteria for cocaine use disorder. They've been using cocaine for many years, around 20 years. And they've been using it for a few times a week. And we can match our cocaine-addicted individuals with our healthy controls on factors, on demographic factors such as age, sex, race, intellectual functioning, but also some things that are part of the phenomenology of addiction, which is depression that is elevated in addiction, and other drug use, such as nicotine smoking. So we are able to match, and we are able then to attribute results to cocaine use itself. So what are our predictions, having described the prefrontal cortex and how important it is to salience attribution, value attribution, and to self-control? So the first prediction is that attention and higher order cognitive functions are going to be impaired in drug addiction, and it is associated with the prefrontal cortical function and structure. Why are we focusing on the prefrontal cortex so much? Well, here, if you look on this slide, you can see that across species type of look at the prefrontal cortex, the white areas, and you can see that in the man and woman, around 30% of the entire cortex is occupied by the prefrontal cortex. So if you compare it to our beloved cats and dogs, it's around, in the cats, it's around 3.5% of the entire cortex that's occupied by the prefrontal cortex. In the dog, it's around 7%. So it doesn't mean that they are not as smart, and especially around dinner time, and very smart to manipulate our own behaviors, right? But it is larger in the human species. Not only it's larger, it's also very variable, and functionally significantly so. So here you can see the view of the brain that focuses on the prefrontal cortex, mid-sagittally, from lateral view, ventrally, and it is subdivided to all these different regions by Brodmann, and we call them Brodmann areas. And the significance of that is that these type of regions have functional heterogeneity. So for example, here to the right in this slide, you can see a region of the prefrontal cortex that's called the posterior medial frontal cortex that has been shown to be involved in self-regulatory behavior. So for example, in identifying and monitoring conflict and uncertainty, error, and in feedback. So this region here is known to be involved and to be important, crucial to our ability to regulate behavior. What happens when the prefrontal cortex is damaged? And I start with the example of Phineas Gage, but I'm going to skip it, assuming knowledge in this audience for this case. And I assume that the red baron is a little bit less well known, but this is an example actually that is even more relevant to addiction. So this is a person who was extremely widely respected for being a very disciplined combat not only a pilot in World War I of a combat aircraft, but actually he commandeered a whole fleet of aircraft, pilot aircraft. And he, similarly to Phineas Gage, sustained a lesion to his frontal cortex. You can see on the left here this hole in his helmet, showing the location of his lesion. But similarly to Gage, he was able to miraculously recover function to such a high level that he was back in the plane. So flying a combat airplane in World War I was not an easy feat, but he was able to do so only to die 9 months after sustaining this lesion by doing something that every disciplined combat pilot knows not to do, which was the inability to stop a behavior that was very disadvantageous to him. So he was so fixated in his pursuit of an enemy aircraft that he did something that he knew that he should not be doing, which was fly very low above anti-aircraft machine guns, and basically was shot down and promptly killed. So this is an example of how a lesion to the prefrontal cortex causes a person to do a very disadvantageous behavior that leads them to their demise. And I think this is a very relevant example to what happens in addiction. So if you test people with those type of prefrontal cortical lesions with general intellectual functioning type of tasks, with learning, with memory, with many, many different type of tasks, you will see no impairment. And actually that's what led the red baron back to the airplane. But you have to use very sensitive and different types of tests. So here, for example, tasks of decision making. And there are many different tasks. And here I'm just showing 2 examples, but I'm going to focus on the Iowa gambling task. It's a game card where you have to basically maximize your game. So there are 4 different types of decks, and you have to choose from these decks to maximize your gain and minimize your loss. So let's say you chose a card from deck number 2, and you win $100. But you also lose a lot of money, and you have a count constantly of your previous gain and your current total. So then you can either keep choosing from that deck, or you can choose from different types of decks. So for example, here at the bottom you choose from deck number 3, and here you win a little bit less. It's only $50, it's not $100, but you don't lose. And healthy individuals, slowly with this task, they learn which of those decks are advantageous to choose from and which are not. And they're able to do so because they're able to give up on a reward, even if it's very, very high right now, because they're thinking about the longer term consequences. And they know that even if I get a higher reward right now, I'm not going to go for it because I'm thinking about the later consequences. People with prefrontal cortical lesions and people with addiction have an impairment in this ability. And they go back to those decks that are already identified as the disadvantageous decks, because maybe this time they will win. Maybe this time it will be a little better. So there is a type of discounting of what happens later on. So this is the bad deck, that's the good deck. Okay, across many different labs now, and across different types of substance abuses, structural MRI has been pretty consistent. And structural MRI, and we're using the magnet, we're quantifying the brain's morphology. We can separate it to gray matter, white matter, to CSF, and we're focusing on the neuronal gray matter. Here we basically quantify each voxel. We are assigning it a probability of belonging to a particular tissue class based on its intensity and information from prior probability images. So here you can see this yellow blob. And what has been shown across these different labs is that when you compare healthy individuals to people with addiction, there is a decrease in the gray matter in this region in people with addiction. So there is a decrease of the integrity, of the neuronal integrity in this region here. And it correlates with the ability to make choices, advantageous choices. So here, the higher, you can see that the lower this type of gray matter in this region here, the ventromedial PFC, prefrontal cortex, the more playing bad decks on that game that I showed you. So the more people have decreases in their gray matter in the prefrontal cortex, the more their choices are disadvantageous if they're drug-addicted individuals. And that relates to the choice and to the behavior of drug seeking. So if this is type of the neurocognitive correlates of making those bad decisions that then culminate and lead to then the continued drug use patterns we see, despite all the catastrophic consequences, this can be an underlying mechanism. In our lab, we showed a very similar pattern in the very similar brain region. And so here in the red spot, again, decreases in gray matter in cocaine-addicted individuals as compared to healthy controls. And this time, it's associated with lifetime years of cocaine use. So the longer, those spots here that you can, those circles are individuals. So this individual used cocaine lifetime more than 30 years, almost 40 years. And they have also the lowest gray matter density in the orbitofrontal cortex. So the longer the lifetime use of cocaine, the lower the gray matter in the orbitofrontal cortex. And this region was uniquely associated with cocaine, but other brain regions that are extremely important to behavior, not only to decision making, but to learning and memory, such as the hippocampus. So the hippocampus is very important to the ability to actually extinguish associations if they are no longer rewarding. So when you learn something and it was rewarding, it's worth repeating it if it is still rewarding. But if it's no longer rewarding, if it's no longer advantageous, no longer relevant, it's actually important to make the dissociation. So that's one of the roles of the hippocampus. And here you can see that you have the longer the lifetime years of cocaine use, and cigarette smoking, and alcohol drinking, the lower the gray matter in this extremely important brain region. So if we can somehow discontinue or diminish or decrease years of drug use, maybe we can somehow offer protection against this type of damage to the brain of people with addiction. So this type of, those are correlations, and we cannot attribute causality. We cannot say, okay, drugs have led to these decreases in the gray matter as a function of lifetime years of cocaine or use of other drug use. So it can be, it's a correlation. It could be that drugs cause it, but there could be also other factors, vulnerability factors that predispose an individual to both decreases of this gray matter in those very important brain regions and excessive drug use. And we do not know that. It's only longitudinal studies in humans that start before a person starts using drugs may shed some light on that question of causality. But we can take a look at what happens if a person is abstaining. So whatever the cause for that, and again, we cannot attribute it to abstinence itself, but it can give us some clues as to dynamic changes in the brain. Is it possible? What's the flexibility of those regions? And here I'm showing you results, again, of gray matter volume in cocaine-addicted individuals at baseline. And this is when they are in treatment or they just finished treatment, and we follow them up for 6 months. So 6 months later, we get them back and we conduct again those scanning and those studies, and we assess the brain regions again. And during these 6 months, people have substantially, significantly reduced their drug intake. And you can see that there is a recovery, an increase of gray matter in the inferior frontal gyrus and in the ventromedial prefrontal cortex in cocaine-addicted individuals who remain abstinent for 6 months. And the important thing is that the same pattern you can assess behaviorally. So only in the cocaine-addicted individuals, you can see in the left, on the bottom left graph, this is the net Iowa gambling test score. So I showed you that Iowa gambling task, and here we are looking at advantageous over disadvantageous decisions. And you can see that at baseline it was lower than in healthy control subjects on the right, but 6 months later in the lighter blue at follow-up, there is a recovery of a decision-making function. And now at 6 months, they no longer look different than the healthy individuals. So not only their brain recovers in terms of its neural integrity, but their ability to make advantageous decisions recovers. And maybe that underlies their ability to remain abstinent for 6 months, which is a no small feat. Okay, so next to the next prediction, and drug addiction is not traumatic brain injury. There are similarities, but there are a huge amount of differences. One of the differences in addiction and what makes it such a unique and amazing disease to study, it is responsivity to context. And attention bias to specific context, usually associated with drug-related cues. So let me show you what I mean by attention bias and how it happens to all of us. So I would like you to think in your head and go through this exercise with me of naming the ink color of these words. So here it would be, okay, color is green, color is red, yellow, and blue, right? It's easy. But what happens if I tell you to do the same thing again, but for this set of words? So for this word, what is the color? Is it blue? Oh, no, it's actually red, right? The next one, it's not yellow, it's green. And then it's actually blue. And then it's actually yellow. So this set of words was much, much harder. If you went through it with me, you saw that your first inclination is to read those words. And what you read conflicts with the color. And if you go through this task very, very fast, then you will be wrong. And because the task of naming the ink, which is the task at hand, competes now with something that's much more automatic and faster, which is reading the word. So you have this type of conflict, you have this bias to the reading. And that has something that's called the color word Stroop effect. It's very reliable in eliciting this conflict. It's very automatic, and it gives a great sense of what it is to resist something that's so automatic. So we can change and we can play with those tasks, and we can change the words. We can use color words, or we can use other words. So here, for example, words that are related to the lecture that I'm having now. So we can color name, but different type of words, and we are supposed to ignore what we read, but we cannot. And then if we continue going, and now this time I chose words that are related more and relevant more to people who think about alcohol and about drinking. And so while all of us are doing the same thing, which is color naming those words, the meaning of those words affects our behavior, and they influence our reaction time, how fast we can do things, and our accuracy. So this is the emotional version of this truth task. So we psychologists like to play with those type of tasks while we are actually scanning the brains of individuals who are performing those tasks. So people can do this type of tasks while they're in the MRI, and now we are measuring their brain activities. So I showed you results of brain structure and morphology, gray matter, and here we're using fMRI BOLD, the blood oxygen level dependent signal. And here, through a process called the hemodynamic response, blood releases oxygen to active neurons at a greater rate than to inactive neurons. So this causes a difference in magnetic susceptibility between oxyhemoglobin and deoxyhemoglobin, or between oxygenated and deoxygenated blood. So again, there is no injection of anything, there is no radioactivity, no IV, nothing is administered. The blood properties themselves are used as the contrast agents, and anybody basically can do those type of tasks while in the scanner. So here people are reading those words. They can be drug-related, they can be neutral, and then they have to press for the color. They also receive money to perform this task. And despite the fact that there are no differences in the engagement in this type of task in cocaine-addicted individuals as compared to healthy controls, we see differences in their brain activation in regions that are essential for this behavioral monitoring, the cognitive conflict resolution that I mentioned here on the left in the dorsal cingulate cortex. You can see in cocaine-addicted in black addicted individuals, reduction in this fMRI BOLD signal as compared to healthy controls. And the same type of reductions are there also for this region, the medial orbitofrontal cortex, I already mentioned it, that is so essential for emotion regulation. So here there are abnormalities at the level of brain function when people are engaged even in this very grabbing, attention-grabbing, drug-related and salient task in people who are cocaine-addicted as compared to healthy control. So they have prefrontal abnormalities when they're trying to manage this type of conflict that is arising based on this type of words. And so in other labs, it has been shown that this type of attention bias to the drug-related context can be managed in some way if you are training people. So here is a study where people were trained, heavy alcohol drinkers were trained at baseline 1 and baseline 2 to ignore this context. So they were playing this type of drug troop, emotional troop type of task, but they were training on responding to the colors of the frame. So ignore what you see, ignore the bottle of whiskey, ignore the bottle of beer, but instead press for the color of the frame. Do something neutral, and do it again and again and again. And the amazing thing is that then at post-training, 3 months later, and even 6 months later, up to several months later, there was not only an improvement on the performance of this task, but there was a decrease in the amount of drinking. So people decreased their drinking from about 60 units to about 50, so 10 units a week, which is pretty substantial. So this type of cognitive training of reducing attention bias, but also other type of trainings of improving working memory, of improving the ability to think about the consequences, can actually have an immediate effect on an individual's ability to resist drugs to a certain extent. And we think it is because of the effect on brain function, and I'll show you that later. Okay, so moving along to the next prediction is that what happens with reward. I spoke about cognitive function. I spoke about attention to drug-related cues. What happens with reward? And this is very important because usually the perception of the drug-addicted individual is this type of, there is an over-focused responsiveness to rewards. And actually this is one interpretation that I think goes in the flow of thinking about addiction as not a disorder of the brain, that there is just hyper-focusing on high and on rewards. But let's see what the data actually shows us. So here we use money, and money is a secondarily generalizable reinforcer. So we're not born with it, born knowing we need it to survive. It's not like food that we are born knowing it. But we can use it in humans because of the learning process associating money with a lot of different types of rewards that happens very, very fast. And we can modulate its level. We can have high monetary reward, low, anything in between, and we can tie it to a performance. So we can really use it to enhance motivation and task engagement in individuals. So here is an fMRI BOLD study, again, in healthy individuals. While they're doing a very simple working memory task, actually to go, simple go, no-go task. Sometimes they have to go, sometimes they don't go, press or not press, depending on the stimulus they see. Very simple, but still demand their attention. And they receive money for every accurate response or non-response. We can compare high money reward to no reward. And those are money, although it's only 45 cents, it actually adds up to $50 just for that task. So in the healthy individuals, we see all these different activations in all these different regions, including the orbitofrontal cortex, including the midbrain. This is deep subcortically in the brain where the cells that actually produce dopamine are located, and dopamine is extremely important to our ability to perceive any pleasure, reward, or to have salience, and to many other type of functions. And we see that in healthy individuals, but what happens in the cocaine-addicted individuals? So if you think, oh, addiction is about reward, then you would expect to see many more activations. But really what happens at this statistical level is that when we compare high reward to no reward, to compare everything a person sees and does, we see no activations in the cocaine-addicted individuals to this contrast. And this is a counterintuitive result. It means there is a decrease in the response of the brain to something that's essential. To even getting drugs, we need money, right? So we performed the study again, this time using EEG that I mentioned before, and here we're focusing on the event-related potentials. So those are positive and negative deflections in the ongoing EEG, but they're time-locked to a certain event. And the events we're measuring here is this simple task. And here I'm showing you results in healthy control subjects, measuring the P300. It's the component, the positive component that develops around 300 to 600 milliseconds after a salient stimulus. And you can see how healthy individuals can differentiate high money, low money, and no money. So there is a clear difference between response to reward in healthy individuals, but it's not there in the cocaine-addicted individuals. And here it is validating our fMRI results. Those are different subjects, an entirely different tool to measure brain function. And again, we see decreases at the cortical level of responding to reward. And this actually is extremely consistent with studies in drug addiction showing decreases in reward responsivity in people who are addicted to drugs. So there are, maybe as a compensation to a supraphysiological increase in the beginning to drugs of abuse, and then there is a compensation and a decrease of response. So does it generalize to other type of rewards and stimuli? So here we are looking at images, not just money. I showed you attention bias to drug-related cues and decreases of responses to those cues in that fMRI task, the Stroop task. I showed you decreases in responses to money. What happens to this type of images? And here in the healthy control subject, again, we're measuring event-related potentials. This time the marker is the late positive potential. It's very similar to the P300. It's tagging salient events. And you can compare those pleasant images to neutral images. So these are neutral images on the left. You have on the left you have the pleasant images, then the horribly unpleasant images, then neutral, and then drug-related images. And if you look at the control subjects, the late positive potential is increased to the pleasant as compared to the neutral images, and we can measure this component at the early and late window, the 400 to 1,000 millisecond, and then the later window. And you can see the same response to the unpleasant images, and nothing is there for the cocaine images, because if you look at the cocaine images, top row on the right, those little bags, what are they? If you're not using cocaine, maybe you think, well, maybe it's sugar or salt, whatever. But then, if you're a cocaine-addicted individual, immediately you can see the response at both time windows to those cocaine-related images. They're definitely not neutral. They're definitely evoking an emotional response. And another thing you can see is that although there is a response to the pleasant and unpleasant images, the response diminishes. It habituates at the later window, the 1,000, 2,000 millisecond window for both pleasant and unpleasant images. So there is a decrease in the emotional responsivity to very powerful salient, both pleasant and unpleasant images in cocaine-addicted individuals, but you can still see the attention bias to the drug-related images in cocaine-addicted individuals. So what can we do about that? And I showed you already, I already spoke about cognitive intervention to try to reduce this type of attention bias by a simple cognitive intervention. I also showed you results of abstinence. If a person is just abstinent for 6 months, there is improvement in the structure and decision-making in addicted individuals. But beyond abstinence and beyond cognitive training, how can we enhance recovery? And we need to think about enhancement of recovery. Addiction is a lethal disorder. So I want to mention briefly substitution therapy. So here, for example, we use methylphenidate. It's better known as Ritalin, basically what we use for children with ADHD. And methylphenidate blocks the dopamine transporters. It increases extracellular dopamine. So it functions very similarly to cocaine itself. It also increases salience of food stimuli and math tasks in healthy individuals. So it bolsters cognitive functions by improving the signal-to-noise ratio and optimizing activity in brain regions that support accurate task performance. And it actually decreases risk-taking and improves inhibitory control in other psychopathologies that affect the prefrontal cortical integrity and function, such as ADHD that I've mentioned, but also traumatic brain injury, stroke, and dementia. So the hypothesis is that methylphenidate will also improve brain and behavior function if given at lower oral dose to people with cocaine addiction. And this type of interference of the binding of the drug to the target, but slower pharmacokinetics. So methylphenidate blocks the dopamine transporter, but it does so at a slower time scale. So it interferes with the function of cocaine. This type of interference has been used and valuable in the treatment of other drug addictions, for example, with heroin and nicotine. So methadone maintenance with heroin. So here is a study you already saw this type of task, the emotional drug Stroop task. But here we are taking cocaine-addicted individuals and healthy controls, and we're giving them, while they're performing this task, either methylphenidate, 1 pill, 20 milligrams, or placebo. And they're performing this task while taking methylphenidate versus placebo. And you can see here at placebo, in the same region where I showed you there was impairment, the dorsal cingulate, you see the cocaine-addicted individuals have lower bold response under placebo. But when you give it the methylphenidate, there is a recovery of function of that region that is so important to the ability to control behavior. And the same pattern of normalization is seen in the ventromedial PFC with 1 single dose of methylphenidate. It was also associated with improvement of function, with reduction in the errors of commission of errors on this task. So with less impulsivity on this task. So both at the brain and the behavioral level, you see an improvement of function with methylphenidate in cocaine-addicted individuals. And we showed the same type of improvement. I'm not showing you again this here because of lack of time, but the same type of improvement of brain function and behavior. We see it also on another task, the classical color word group that I showed you before, and also during resting state functional connectivity, so between regions of the brain and networks of the brain. And okay, so now I showed you, I hinted at several possibilities for improving function in drug-addicted individuals, but what does really happen with addiction? What really happens is that most people don't even seek treatment. So these are out of the 20.2 million who need treatment for drug or alcohol abuse, 95.5, almost 96%. So the vast majority don't get treatment. They don't need they don't have the feeling they need treatment. So maybe it is because addiction is stigmatized. It's hard to admit it. Maybe because people are in denial or deception, and that's something that has been attributed to addiction quite a lot. But we actually think that it may also reflect an impaired awareness and impaired self-insight and self-knowledge and impaired knowledge of the severity of your own disorder. And that can be attributed to underlying neural dysfunction that also affects motivation and self-control. So how do we assess this type of self-knowledge, this self-awareness? It's such a fleeting thing. It's so hard to assess. It's definitely not something that we can assess necessarily in animals. So it's uniquely human, this type of self-awareness. So here are the tasks that we modeled based on the Iowa gambling task I showed you before. Here we have, again, DEX, but this time the goal is not to maximize gain. There is no gain here at all. It's just that you choose to look at what's going on this deck. Oh, here is a pleasant image. Beautiful, nice image, and I'm going to look at it for a few seconds, enjoy it vicariously a little bit. And then I can either choose, again, from that same deck, or my novelty can lead me to choose from another deck, novelty seeking. And I click on this one, and here is a person either smoking or imitating smoking a crack. So then I can go back, and I can choose from these decks. I can go back and choose from the first, second, third, or fourth decks, and I can make my choices. And using this task, we showed that even when the contingencies behind those decks are not clear, and we actually make this task to be that so that most of one deck is mostly pleasant images, the other one is unpleasant, the other deck is mostly neutral images, and the fourth one is crack cocaine type of images. But still, the contingencies are not entirely clear. It's not 100%. Despite this type of implicit contingencies, healthy individuals show a clear preference to look at pleasant images, and cocaine-addicted individuals have a preference to look at cocaine-related images much more so than healthy control subjects. And the choice to look at those drug-related images over the pleasant images is associated with concurrent drug use outside the lab, but also previous drug use, and it predicts 6 months drug use later. So just playing this simple game of what is it you want to look at can predict abstinence and outcomes 6 months later. But I'm showing you this task because I want to talk about self-awareness. So we ask people after they complete this task what kinds of pictures do you think you chose to look at most often? And the split between what they say and what they do, this is our measure of self-awareness. So you can do one thing. When I ask you, what is it that you did? You can tell me something else. Or you can be consistent with your choice. So this type of match versus split between what you say and what you do, for us, is an indication of self-awareness. So in healthy controls, most of them are very accurate in reporting what is it that they chose to look at. But people who are current cocaine users, really their performance is below 50%. It's like they have a mismatch, and actually most of them have a mismatch between what they did and what they think they did, and it is driven by viewing those cocaine-related images. And people who are abstinent, they're in between. So when we look at this type of people who are impaired on self-awareness, so they have that mismatch between what they say and what they do, we can see that this region here, the rostral cingulate part of the prefrontal cortex, is lowest in response to error in the people with impaired self-awareness. So we measured this self-awareness outside the scanner with this task, and we can see that this subgroup of individuals, of cocaine-addicted individuals, have also the lowest response to errors, to error monitoring with this, in this region, and it correlates with cocaine use. So the lower this type of pattern in this region, the higher is their cocaine use in days per week. And the same region, if we look at it now structurally, if we look at the gray matter and we quantify it, the same group of people with this mismatch between what they said and what they did, and with the lowest response to error in this region, also show the lower gray matter in the same region. So this may be a neurocognitive type of mechanism that underlies this inability to correctly monitor your own behavior in response to either error or in response to your choice. And it looks to be both at a functional and a structural level, and it correlates with actually frequency of cocaine use. Okay, so let me summarize all the amount of data I showed you and some of the take-home messages. So I showed cognitive impairment and associated with prefrontal cortical integrity and function in individuals with cocaine addiction. And I spoke about the effect of chronicity of use, but also effects of what abstinence and treatment. I spoke about attention bias to drug-related cues, focusing mostly on cortical mechanisms. Subcortical mechanisms can be actually different, but I didn't have time to go to it today. And I spoke about modified neural sensitivity to monitor, reward, and non-drug stimuli. And there are similar results from other labs and other drugs of abuse. It is also consistent that decreases response to reward in addiction is consistent with decrease in prefrontal cortical activations to other non-drug reinforcers and salient reinforcers, such as erotic videos, for example, in cocaine addiction. It also is consistent with the fact that the value of the drug reward is increased and overpowers the value of non-drug reward, potentially because of the supraphysiological stimulation by drugs and the adaptations that happen at the neural level to those type of supraphysiological stimulations. And if we have this type of compromised sensitivity to reward, then decision-making is necessarily impacting. What is trading a car for a couple of cocaine hits? These type of things become blurry. They're not as clear depending on the state you're in. And it correlates with depression, anhedonia, of course with impulsivity. I talked about impaired insight. So how do we treat people? What can we do to the subgroup of individuals whose insight and self-awareness is impaired? It's clear that maybe we can help them in different ways to the people whose insight and self-awareness was not impaired, it was intact. So I showed you a subgroup of cocaine-addicted individuals whose self-awareness is intact, and we think that their treatment is probably going to be different. It also is very important to, all our thoughts about self-awareness and insight is just summarized here in this paper here, so I'm showing that. And it is extremely important for relapse prediction. So if we can look at those impairments and abnormalities at baseline, and we can use them to predict treatment outcome and response to treatment, maybe we can, in some people, and using different markers in other people, maybe we can intervene in an individually tailored way at the right time. There is a balance in your brain. I spoke very much, I focused on the prefrontal cortex, but the prefrontal cortex is very much interconnected with many, many regions of the brain. And really, it's all about systems and connectivity between different systems supporting different functions. And many things can be threats to self-regulation and to this type of intricate and complex patterns of intercommunication in the brain. So cue exposure and lapses, negative mood, resource depletion, drug consumption, potential brain damage, all of this can imbalance this delicate type of dance in the brain between the prefrontal control and impulses in the brain. And what we would like to do is actually to put it back in balance, such that a person can find their own ability to self-regulate again. And I'll end with this slide showing the iRISA model again of drug addiction, and mentioning that people, again, the cycle of addiction that has not only states of intoxication and bingeing and craving and withdrawal, where a person wants to either go for the drugs or there is a lot of conflict around it, but there is also a healthy state. In every person who is addicted, there is a healthy state where non-drug related functions are predominant. And what we want to do as treatment providers, we want to give people tools, as many as we can, to go back again and again to that healthy state in a way to protect their behavior and brain function and structure. So let me just finish by thanking my group. This is the Neuropsychoimaging of Addiction and Related Conditions group now at Icahn School of Medicine at Mount Sinai. I presented work by Mohammed Parvaz, by Scott Muller, by Nelly Alea Klein, but it really takes a huge group and many years to complete all these studies. And I'll end with a slide of the Brain Imaging Center at Sinai and thanking our funding agencies, mostly the NIH. Thank you. And NIDA.
Well, thank you, Dr. Goldstein, for that fantastic presentation. That was really fascinating. As a quick reminder for our audience on how to submit questions, simply, if you have any questions, type them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as time permits. So Dr. Goldstein, you have a question right now, and it's from Andrew at CSU Long Beach, who asked, as you gave an example with opioids, cocaine isn't a prescribed drug. Which amount is used to become addicted to it?
Oh, it's really different. For different people, it's very different. It's not, the higher the amount, the worse it is. The faster it goes to your brain, the worse it is. So more people become addicted, for example, to crack cocaine than to sniffing cocaine, although people are also, they also know there is addiction to just sniffing cocaine, absolutely. So it's a higher amount and faster delivery to the brain is associated with more addiction.
Great, interesting. Another question from Andrew again. He asks, is gray matter density from recovering addicts able to increase back to normal levels of gray matter density, I guess, in normal people?
So what I showed in our results, I did not present the healthy control levels, but yeah, there was definitely a normalization to the healthy control level where there was no difference between the two measurements. And so yes, there definitely is. Some brain regions are more resistant to change, and some damage takes longer to recover. And so it really differs by the brain region and circuit. But the short answer is yes, there definitely is a lot of recovery.
Interesting. We have a question from a member who I don't know if this question you can answer, but I'll ask anyways. He wants to know your best advice to reduce teen suicide risk.
This is well beyond my area of expertise. I spoke about addiction. There is a lot of drug use and drug experimentation in teens. This is a very vulnerable period, and people definitely, and there is a lot of comorbidity between drug use and also mental health issues in the teen years. It's really a vulnerable time period, and more vulnerable to some people than others. And so my advice to reduce teen suicide risk is to be very much on top of your teen constantly at all levels of family, home, school, a lot of one-on-one daily, and treatment, immediate treatment of symptoms and then of diseases. There are a lot of diseases that start at teenage, depression, anxiety, and kids will self-medicate. So as parents, we need to offer them medication that actually will not harm them. So psychologists, psychiatrists, all of the above, treatment is an intervention is really essential.
I see. And if the audience wants to learn more about teen suicide risk, I think there's a presentation after this one that's presented by Dr. Jay Gould that focuses more on that if they wanted more information. And so we have time for one last question from Ivana, who asks if one suffering from chronic pain can become addicted to cocaine consummation.
Anybody can become addicted to cocaine, and maybe people with chronic pain are more vulnerable. It's a chronic stress type of situation. So you definitely have heightened stress, heightened negative emotionality. In animal models, I think you do see that type of crosstalk. But just being in chronic pain, it's not the whole picture. It's the vulnerabilities that add up together. And it goes back to the way I started this presentation by defining addiction. Just being in chronic pain and getting opioids for treatment of pain will not make you addicted to opioids. So we tend to under-medicate. That is my opinion. And being in pain or in stress or in depression, those are highly vulnerable. They put individuals in highly vulnerable places, and I highly recommend treating those primary disorders because people will eventually treat themselves, and sometimes this will not be by the right drug.
Very interesting. So thank you once again for such a fantastic presentation. Do you have any final comments for us before we part?
No, thank you very much for having me, and thank you for wonderful questions.
Yeah. So I'd like to thank the audience for joining us today and for their interesting participation. And as a reminder, this webcast can be viewed on demand through September of 2016. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, see you. Bye-bye.